Okay, finally in this third video, we're going to finish looking at the process of translation. We now understand how tRNA is made. Now we need to look more carefully at the ribosomes, because both of these are used in the process of translating an mRNA sequence into an amino acid sequence, which is otherwise known as a protein. And remember, this protein is the functional unit that's based upon the DNA sequence that corresponds to a gene. So we have this relationship between a gene and a protein. Okay, so let's look at how the ribosome is used in translation. So first, what a ribosome is it is made of proteins, but it also is made up of what's called ribosomal RNA. So this is RNA that is made as part of the process of transcription, but then it's done. After transcription, that is the unit. We don't need to translate it. That unit of RNA is used in the ribosome, and it's called ribosomal RNA. Now, the thing about the ribosomal RNA genes that's kind of interesting is that they are evolutionarily conserved. That means that across species, if we compare a human's ribosomal RNA to a mouse or a rat's ribosomal RNA, in this picture we can see that those nucleotide sequences are nearly identical. And there's certainly parts of it that are more heavily conserved than others, but across all of these, we're seeing the exact same sequence of nucleotides, human, mouse, and rat. And if we compare these sequences from the vertebrates to some of these bacterial species, such as E. coli, we'll see that they're also heavily conserved, but only in certain spots. So this picture kind of shows us, look, there's some variability in the red regions here, but it also shows us, look, these huge yellow regions are 100% conserved even as you move from prokaryotes to eukaryotes. So the rRNA subunit genes are evolutionarily conserved. They're going to be very similar among species. So the ribosomes are going to be a little bit different, though, between the bacteria, the prokaryotes, and the eukaryotes. And then within eukaryotes, there's going to be different types of ribosomes in different cellular compartments. Remember, the mitochondria and chloroplasts are kind of thought to have originated from bacteria with that endosymbiotic theory that we talked about way back. And the mitochondria and chloroplasts, they do have ribosomes, but their ribosomes are more similar to those of the prokaryotes, which kind of lends to that endosymbiotic theory of the semi-autonomous organelles, mitochondria, and chloroplasts. Okay, so ribosomes in these species will all contain what's called the large subunit and the small subunit. They are different between bacteria and eukaryotes. This is giving us a unit of weight or size for these, and so there's going to be some differences there. There's differences in the number of proteins and the number of RNA. Without getting too lost in these details, both prokaryotes and eukaryotes, the ribosome will contain a large subunit and a small subunit. And we're going to be seeing that in each of these pictures. Another thing that I want to connect is, remember the nucleolus? The nucleolus doesn't get a ton of attention, but here's the one place where it really should get some attention. In eukaryotic species, remember, they have a nucleus, and inside the nucleus is a dark spot, which is called the nucleolus. And the nucleolus really is just a dark spot. It does not have a membrane around it. It's just a dark spot in the nucleus. What the nucleolus is, what this dark spot is a ton of ribosomal RNA. So this is the region of the nucleus where the ribosome is assembled. Now the ribosome, it's not just ribosomal RNA, it is also proteins. And those proteins are made in the cytosol and then transported back to the nucleus, to this region that becomes a really dark region because it has all these proteins and RNAs. And in the nucleolus, the reason it appears so dark is because it has a big collection of the RNA and protein, and it's making the ribosomes. So that's what the nucleolus is. It's really where we make the ribosomes that are used in the cell. And then after these subunits are made, they do have to leave the nucleus because ribosomes are going to be found out in the cytosol. And that makes sense because we also talked about how the mRNA leaves the nucleus to go find the ribosome out in the cytosol. Okay, so let's have a look at what the ribosome is. Remember I said it has a large subunit and a small subunit. So we're seeing that here in this picture. This is the large subunit, kind of up top. So that's the large subunit. And this is the small subunit, kind of down below. Now the thing about the ribosome is that it's going to interact with the RNA. So here's the mRNA. It's kind of loaded onto the ribosome in this picture. And then the ribosome is going to have 3 sites that we're going to talk about. They're called the peptidyl site, the aminoacyl site, and the exit site. And they're in the order E, P, A. So that's going to be our exit site, our peptidyl site, and our aminoacyl site. And we're going to watch as the mRNA kind of threads through here and moves along. We'll see different things happening in each of these 3 regions of the ribosome. Also in this picture, you're seeing the tRNA. tRNA, remember, is bringing in the amino acid that corresponds with the anticodon that corresponds to the codon of mRNA. So this is going to be important. So when we talk about translation, 3 things have to assemble. We have to have the mRNA load onto the ribosome, and then we have to have the tRNA, many different tRNAs come and bring in the appropriate amino acids. Let's look at how this works. So we're going to see 3 stages of translation. These are going to look a little bit like the 3 stages of transcription. In fact, they're named exactly the same: initiation, elongation, and termination. In the first stage, we're going to see everything is assembled. Then we're going to see the mRNA chugging along, and we're going to watch as translation happens. Then finally, we need to have reach the stop codon and have everything disassemble and have that protein released. This picture shows those 3 steps. Initiation, where everything is assembled. So here's our ribosome, here's our mRNA, here's our first tRNA, and here's a large subunit of the ribosome. So this is step 1. This is initiation, where everything is assembled. Then this begins moving along. So the ribosome moves along the series of mRNA, and this is the elongation stage, where we're going to be bringing in various tRNAs and connecting the amino acids to a growing polypeptide. Then step 3 will be termination. This is where we reach the stop codon and everything's going to fall apart, and we're going to have that completed polypeptide. We're going to have the ribosome is reusable. All the tRNAs can be recharged and they can be reused as well. I'm going to try to show you this here in a series of steps and kind of talk you through it, but I've also linked this video into Brightspace, and this video kind of shows everything in action, which maybe gives you a better view. But let me try to walk through the separate steps. So step 1 is initiation, and here we're actually looking at what happens in prokaryotes. In a second, I'll talk about what's slightly different in eukaryotes. But first, let's just look at what happens in the prokaryotes. So this is the small subunit of the ribosome. You see that the ribosome, remember, it has some rRNA, so we're kind of seeing that here in red. And the ribosomal RNA is going to correspond to the ribosomal binding sequence, which is part of that 5' untranslated region that we talked about existing in the mRNA. This is upstream. It's 5' upstream of the start codon. So that start codon, AUG, is not necessarily found at the end of the mRNA. It's found a little downstream of what's called the ribosomal binding sequence, because we got to use this ribosomal binding sequence to kind of load the mRNA onto the ribosome. So the rRNA of the ribosome interacts with the ribosomal binding sequence of your mRNA to kind of get things docked and lined up. Then our AUG, or our start codon, can be in frame to begin or initiate the translation process. So the AUG is our first codon, and so it kind of gets loaded on and lined up so that our first tRNA can bind to it. Our first tRNA is going to have the anticodon that matches the start codon. So the start codon is always AUG, and that's going to correspond, the complementary pieces for each of that, A is complementary to U. Remember, it's not T. T is replaced by U, and we're talking RNA. The U of the start codon corresponds to an A, just like a T would, but we have a U instead of a T, so the A and U are complementary, and still C and G are going to be complementary. So our start codon, AUG, is complementary to the anticodon in our first tRNA, which is charged with the first amino acid, and this would correspond to the codon as called for in that genetic code chart, which is actually going to be methionine. And that's going to be the same always. Every protein's first amino acid is actually methionine because of the start codon is always going to be the same. Okay, then once we have that then the large ribosomal subunit kind of pops on, and that gets this. Now we have our ribosomal subunit assembled. We've got our RNA threaded in there. We've got our first tRNA in place, so we kind of snap on the top of the ribosome, and we've got this complex fully formed. You'll recognize in this picture we've got the three domains: the E, exit domain, the P, the peptidyl domain, and the A, the amino-acyl domain. So everything is now assembled. We've got the start codon in place. We're ready to start chugging along. The ribosome is going to start moving along this sequence of mRNA, and it's going to move from the 5' end towards the 3' end, and that's a rule. It always goes that way. Okay, so this was initiation in prokaryotes. For eukaryotes, it's basically the same, but remember that 5' cap that we talked about? The mRNA has to be capped, spliced, and tailed before it leaves the nucleus. This 5' cap had a modified guanine on it, and that's actually recognized by what are called cap binding proteins that are going to help the mRNA bind to the small ribosomal subunit. So that ribosomal binding sequence is going to be a little bit different for eukaryotes, but we still get it loaded in. There's also going to be what's called a Kozak sequence, which is this well-characterized sequence of a lot of G's and C's that are going to be around the start codon. And we know that this Kozak sequence, we've studied it carefully, and we found that this is really important. If you modify it, even though it's not translated, and it kind of seems like, well, why is it so important? We know that it is really important. The small subunit needs this Kozak sequence, or it might actually skip the start codon if there's something different there. And so this is things that we've discovered experimentally, that the sequences around the start codon are actually really important in the eukaryote. Okay, but basically it's the same. We got to get the mRNA loaded onto our ribosome, and then we're ready to chug along doing this process of translation. The second phase is called elongation. This is where the ribosome is moving along the RNA, bringing in the next tRNA. But let's look at what happens with these three separate regions, the E, the P, and the A. The peptidyl region, let me start with the amino-acyl region. The amino-acyl region is going to kind of have expose these three nucleotides for the codon, and the anticodon of the tRNA is going to load into it and bring the amino-acyl that it's charged with. So that's why this is called the amino-acyl domain. It's kind of the loading domain. We're going to load on the next tRNA that's got its little amino acid hanging out. In the peptidyl domain, this is going to be, initially it'll just be that start tRNA, the one that's charged with the methionine that matches the AUG. But this is kind of shown that this process has happened a few times, and so we're not seeing the very beginning. In the peptidyl domain, this is where we have a polypeptide that has already grown in our picture. If we had been at the very first, it would just have that methionine on it so far. And then what we're going to have happen is we're going to add this next one to it. So what happens if we kind of move to this next picture? What happens is the peptidyl domain is going to transfer its whole chain to this new amino acid. So the whole growing peptide gets transferred to bind to. So what we're doing is we're binding this new amino acid to the existing amino acid chain. So those guys become connected here. And this whole thing gets transferred on top of the incoming amino acid. Then everything shifts over. Everything shifts over by 1. And what happens is this guy, who has now released the whole peptidyl chain, is going to move over here and be released. And so that's what's happening here. It's being released. And now in the middle, we have this guy who was here is now moved to the middle, and it now has that peptidyl sequence to it. It now has that growing peptide attached. And in comes a new guy. So the whole thing, you can either think of it as the ribosome moves this way or the RNA moves this way. I think sometimes you'll read it both ways. I'm not sure which is more correct to say because they're both moving relative to one another. But here it's written the ribosome moves to the right. And so as the ribosome moves to the right, we kind of line up new things in each of these 3 domains. It's really difficult for me to kind of draw this and illustrate it, so do watch that video that I linked in that kind of shows these guys all moving, and I think that'll give you a much clearer picture of what's happening. But essentially, the process is the tRNAs are lining up with the mRNA according to matching codons and anticodons. And then each tRNA is bringing in the appropriate amino acid so that this sequence of nukes, the sequence of codons, is going to determine the sequence of amino acids in the growing amino acid chain or the growing polypeptide chain. Okay, as we keep moving, we're eventually going to get to a stop codon. If you look back at the genetic code chart, you'll see that there are specific codons which are called stop codons. And that means that there's not a tRNA that's going to be charged with an amino acid that matches this codon. The stop codon is actually only going to bind to what's called a termination factor or a release factor. And so it's not a tRNA charged with an amino acid that binds here, it's the release factor. It's the only thing that can match that stop codon. And as soon as that release factor binds to the stop codon, well, everything is released. The polypeptide, it can't be transferred to that release factor, so it is released. And then the whole thing will disassemble. And when it does, you now have the ribosomal pieces can be reused, the termination factor or the release factor can be reused, and then we now have this polypeptide is now done and formed, and we could talk about how it folds or where it goes, but we've made it. We've made this amino acid sequence, which is a polypeptide. The mRNA is actually intact, and it could be reused as well. There's no reason it couldn't. mRNA, though, is unstable, and it may not be reused very many times because there's going to be various enzymes and things that could degrade it within the cell. But it could certainly be reused as well and make a new polypeptides chain if it just kind of reassembles with the ribosomes. So the big idea for our ribosome is what it did was it provided these framework in order for each of the tRNA to bind and keep transferring this polypeptide to a growing or to a new amino acid in order to lengthen the polypeptide chain until we form this long series of amino acids, which is called the polypeptide. And as I said, this could then be folded into a protein, and this will be, depending on what the sequence was, what the gene was, this protein will have various jobs, such as it could go off and be a transcription factor. It could go off and be a polymerase. It just depends on what the original gene was, what this protein is. But this is our final product. We've now made a protein.